Hello, class. So the next section that we're going to work on is the ear. And so we'll start off with the anatomy of the ear. Right, so the ear is what's responsible for helping us to hear, just like the nose for the smell. Nose has other reasons, other benefits as well. We know the nose can help us with also breathing, getting oxygen into our bodies, but as far as special senses are concerned, nose does the smell, tongue does the taste. We've talked about the eye, which helps us to see. So we've talked about those three thus far. Now we want to get into hearing, into the ear, and continuing on with the special senses. Everybody knows the ear is the structure, and it has its own, sort of original shape. We don't really see the shape of the ear anywhere else in the body. Its anatomy is designed to help out the physiology. The form works with the function, and it's divided into three regions: the outer ear, what you see with your eyes. Then we have the middle ear, and then we have the internal or the inner ear. Each region has a role in hearing, but only the inner ear is associated with the vestibular sense, which is important to equilibrium. All right. If I touch this part of your ear, you won't knock you off balance. But in the inner ear, we have not only the ability to hear, it helps with our hearing, but also with our balance. All right, so this is the structure of the ear. We're all familiar with the sort of the outer ear. We try to keep it clean when we take showers, when we have buildup of ear wax, which we'll talk about the purpose of that. We use Q-tips and things like that to clean out our ears. That's primarily dealing with the outer ear. We see this external auditory canal. Sound is entering into our middle and inner ears. The ear lobule, notice that's where people normally pierce. If they're going to have pierced ears, that's one of the sections that they pierce. We'll talk about that. It's soft tissue there, so it's easy to pierce and to put beautiful little diamonds and things like that. But then we'll get into the middle ear, and then the inner ear. And you see in the inner ear, we have this structure called the vestibule. And we also have the cochlea, vestibule and cochlea, and then the nerve, the cranial nerve is cranial nerve number 8, vestibulocochlear nerve. Okay, just some things to notice. We'll talk about all these structures. So the outer ear, external visible region of the ear. We have the auricle, elastic cartilage, with the exception of the lobule. So it's elastic cartilage. And then we have the lobule here, the fleshy lobule, which is covered with skin. Helps funnel sound into our auditory canal. So sort of acts like a funnel. If you've ever seen a funnel, sort of it's big, gets smaller, helps sort of capture sound. The external auditory canal, the canal that goes into our ear. So first, like getting into the ear, slightly curved tunnel through temporal bone, terminates at the tympanic membrane, right? Lined with modified sweat glands, ceraminous glands. We talked about these when we were in chapter 5. They secrete ceramin, which is an earwax, a waxy yellowish brown gray substance. That earwax is going to lubricate and also help waterproof your canal and tympanic membrane, right? Also traps and removes debris before it gets into your tympanic membrane. We don't want dirt and debris getting into our ears. It keeps this area lubricated and it helps to waterproof it, right? We can go underwater. It's not necessarily the most comfortable thing for your ears, but we can go underwater, and not only can you hear underwater, but your ears are still protected. Right. So remember that is the ceraminous glands, which are modified sweat glands. If you want to go back, you can go back to chapter 5 and review, chapter 5 and review that. Yeah, chapter 5, integumentary system. Okay, and that is the structure of your outer ear. Right. The auricle, cartilage, lobule, external auditory canal. And then it ends at the tympanic membrane, thin sheet of epithelium and connective tissue, 2 tissue types, multiple tissue types, separates outer and middle ear regions. The tympanic membrane cone-shaped with apex or tip pointing into the middle ear. So it's a cone-shaped structure. I need a cone with the apex pointing. So it's a cone with the apex, this end pointing into your ear, connected to tiny bone ossicle, called the ossicle, enables energy of sound waves to reach your inner ear. Now remember, with taste, we wanted to convert, the different chemicals that we put into our mouth, the food, whatever we put into our mouth, convert that into a electrical signal. With sight, we want to convert light waves, photons, light waves into electrical signals. With the smell, it's more chemoreceptors again, convert the odorants, the things that we breathe in, convert those from chemical to electrical. With the ear, we want to convert sound waves. We can't see sound waves, but they cause a certain vibration. And we want those sound waves, that vibration caused by sound, which is a mechanical vibration, if you think about it. And we want to convert that into an electrical signal that can be sent to your brain, processed by your brain. And your brain can say, oh, I'm listening to Professor Fisher teach me about the ear. Right, the middle ear. Let's talk about that. Middle ear is hollow, air-filled, mucous membrane. Again, we have sort of a mucus. Remember the ceraminous in the outer ear chamber, in the canal, outer ear canal. So we have hollow, air-filled, mucous membrane lined chamber within temporal bone. So the tympanic membrane forms the lateral boundary and wall of inner ear defines medial boundary. So we have the tympanic membrane on one side and the wall of the inner ear on the other side. And so that defines the tympanic membrane is the end of the outer ear. And then we have a wall which of the inner ear defines where the inner ear starts from that point on. So in between that, we have what we call the middle ear. Here are some of the structural and functional features. Pharyngotympanic tube. These words can get a little tricky. Now that's a word I wish they would show up on the spelling bee. Pharyngotympanic. That's not a word people use very often, obviously, but it's the auditory tube. Connects the nasopharynx, which we're going to learn about the nasopharynx when we discuss the pulmonary system. And we discuss the structures leading from the mouth all the way to the lungs. Also the nose as well to the lungs. We discussed the nasopharynx. So it connects the nasopharynx with the middle ear. Your ear has a connection with your with your airways. It helps equalize the air pressure on both sides of the tympanic membrane. Have you ever noticed when you are flying and you're landing, you may need your ears to pop, or if you go up as an elevator, to the 30th floor, and elevators move fairly quickly, you may feel the need to like swallow or breathe in order to you feel pressure like on your ears. That's why swallowing or breathing can help relieve pressure on your ears, because there's a tube that connects your nasopharynx, nose, pharynx, so back of the nose into the throat with the middle ear. Right, helps equalize pressure on both sides of the tympanic membrane. Pressure difference between chambers reduce ability of the tympanic membrane to accurately vibrate. Right, so if there is a pressure difference, like when you go in an elevator or fly on a plane, things like that, it can cause you not to be able to hear as well. You'll feel like you're like something is stuffed in your ears almost, because the tympanic membrane won't be able to vibrate as it normally would. You also have the auditory ossicles, 3 tiny bones within the middle ear. They're named by their shapes, connected to one another at the synovial joints. We haven't talked about joints yet, but joints are in the chapter when there's a whole chapter on joints. Form a bridge that links the tympanic membrane to outer wall of the inner ear. Right, the 3 are the malleus, which is shaped like a mallet or a hammer. You'll have the incus, which is anvil shaped, is the midpoint. And then you have the stapes, which stirrup shaped, connected to oval membrane and medial chamber wall that covers oval window. Your oval window is this boundary between your air-filled middle ear and fluid-filled inner ear. And these ossicles amplify and convert incoming sound waves in air into fluid movement. All right, so we've already started sort of transducing sound. Amplify and convert incoming sound waves. That's the point of these ossicles, these 3 small bones. Amplify and convert incoming sound with air into fluid movement. Right, then we have 2 tiny skeletal muscles which attach to the ossicles. We haven't talked about skeletal muscles in detail yet. We've mentioned muscles in the chapter on tissues, but we know skeletal muscles are muscles that attach to the skeleton. Right, skeletal muscles are voluntary and striated, remember? All right, so we have the tensor tympani muscle originates in the wall, the pharyngotympanic tube inserts on the malleus, tightens tympanic membrane by pulling it, and the stapedius muscle originates in the posterior wall of the middle ear, inserts on stapes, reduces movement of ossicles as a unit. Both muscles are components of reflex, prevent damage from loud sounds by reducing movement of tympanic membrane and ossicles, right? Because a loud sound will cause them to vibrate too much. So these muscles, as a reflex, so when you hear something that's too loud, normally you may put your hands over your ears, but your ears actually have ways to help prevent themselves from being damaged by reducing their own movement, the vibration of these ossicles, of these bones in the ear, if the sound is too loud. And this is the diagram here of the middle ear. Right, so we have the tympanic membrane, that's the end of your outer ear. And then we have the malleus, the incus, and the stapes. These are the 3 bones here, which help transduce the sound from the vibration into fluid movement. Okay, that is your middle ear. Otitis media, inflammation of the middle ear cavity, one of the most common infections in children. Ear infection, almost always preceded by upper respiratory infection. Remember, your ear and your respiratory canal are connected. Nasopharynx is connected to your ear, your middle ear. So when you have an infection in ear infection there and a nasal infection, those can be very, very much in common. You'll have ear pain, fever, impaired hearing, irritability. So I just wanted to highlight that. The pharyngotympanic tube can act as a conduit for pathogens, and that's what I was just talking to you about. So pathogens, bacteria, viruses in the nasopharynx can reach your middle ear. So you have bacteria, viruses enter your mouth, and they can reach your ear by this passageway. By the pharyngotympanic tube, right? Inflammation causes fluid to build up, tympanic membrane bulges, it becomes painful. Babies who get ear infections oftentimes are very, very irritable. I'm sure some of you may have experienced that. This is showing you a photo, healthy tympanic membrane with acute otitis. And here's a tube inserted into tympanic membrane via myrigotomy. Notice when you have the ear infection, it's red, it's irritated, it's not good. Children are more susceptible to this because their pharyngotympanic tubes are much shorter, more horizontal, and it's treated with antibiotics. We treat ear infections with antibiotics. Okay. Now let's get into the inner ear. The inner ear is after you get past the outer ear, the tympanic membrane, the three bones, then we get into the inner ear cavity within temporal bone. So with inside your temporal bone, remember when you think temporal, think of the section of the brain. We've also talked about the temporal region of your brain. That's around the region where your temporal bone is. And when we talk about the skeletal system, it'll be even more clear. When we talk about the skull, it's a cavity within the temporal bone subdivided into three regions: cochlea, vestibule, and semicircular canals. Right, so we have this bony, membranous labyrinth. Each labyrinth is a network, serpentine pathways composed of either bone or membrane, respectively. Right, so this bony labyrinth, open passageways that make up the inner ear chamber, and we have this membranous labyrinth. Remember, labyrinth is sort of like a maze. As a membranous labyrinth is the membranes that line the bony labyrinth and contain a substance known as endolymph. Endolymph is a fluid, right? Think of the lymphatic system, which we haven't really discussed in this class, but we will. But endolymph is a fluid in which potassium ion concentration is greater than sodium ion concentration. So very similar to intracellular fluid, the fluid between your cells. You also have perilymph, fluid between bony and membranous labyrinths. Sodium ion concentration is greater than potassium ion concentration, similar to extracellular fluid. Then we have both fluids play a role in transducing sound waves. Sorry, intracellular fluid in the cell, extracellular fluid outside of the cell. I said that backwards. right? So endolymph is like the fluid in the cell, perilymph, the fluid outside of the cell. Both fluids play a role in transducing sound waves. Remember, that's the goal. We want to capture that sound for it to come into our ears and to be transduced, converted eventually into an electrical signal that can be sent to our brain, right? That's the goal of the ear. That's the whole purpose of all of these structures combined, right? So we have these ossicles, these bones. So we first of all, we capture the sound. Auricle helps capture the sound, goes into our ear canal, tympanic membrane, and connects it with the ossicles, start converting the sound from a vibration to a fluid movement, right? And then we get into this inner ear part, right? So it's just helping to try to put it all together. So these fluids in the inner ear, endolymph, perilymph, play a role in transducing those sound waves. Now we've converted it from a mechanical vibration to a fluid. It's still mechanical, but a fluid movement in this fluid, transducing sound waves and head movements into electrical signals. All right, so we have the vestibule located medially to the middle ear, medially towards the center, features membranous oval window, houses the utricle and saccule, two parts of the membranous labyrinth, endolymph filled chambers, and it conveys stimuli about head tilting and linear movement, head tilting, linear movement via neurons of vestibular division of the vestibulocochlear nerves, your cranial nerve 8. We have the semicircular canals, contain 3 tubes of the membranous labyrinth, anterior, posterior, and lateral semicircular ducts, oriented at right angles to one another, can detect rotational movement of the head. So rotation of the head, we can detect that with the semicircular canals. Also have the ampulla, enlarged bulb at base of each duct, contains receptor cells innervated by the vestibular division of cranial nerve number 8, your vestibulocochlear nerve. All right, so this is what we've talked about so far. We have the temporal bone, which this is cut into the temporal bone. We have semicircular ducts and the semicircular canals here, the vestibule, the utricle and the saccule, the round window, and we will get into the cochlea, the vestibular nerve here. So that's a lot about, rotation, turning, movement of the head. Notice that that's the vestibular part of the vestibulocochlear nerve. Now, what's the other half of that? So we see where sort of our balance comes from, being able to detect rotation, movement of the head. Cochlea or cochlea, depending on how you pronounce it, features membrane-covered opening called a round window, separates the middle and inner ear. Cochlear duct, spiral-shaped segment of the membranous labyrinth, connected to saccule, filled with endolymph. Cochlear ducts twists around screw-shaped bony core, then terminates at tip of cochlea. Continuing with the cochlea, we have the scala vestibuli, chamber within the cochlea, superior to the cochlear duct, continuous with the vestibule near oval window. We also have the scala tympani, chamber within cochlea, inferior to the cochlear duct, terminates in vestibule near round window. All right, so the structures inside the cochlea. Both chambers surround the cochlear duct, between them, continuous with one another at tip of the cochlea. So just chambers in the cochlea. And we also have the vestibular membrane, a sheet of epithelium. Remember epithelium, chapter 4, forms boundary between the cochlear duct and scala vestibuli, separates endolymph from perilymph, maintain ion homeostasis of each fluid. One has more potassium than sodium, one has more sodium than potassium. Cochlear duct and scala tympani are separated by this basilar membrane, composed of collagenous connective tissue, connected to small shelf of bone projecting from bone labyrinth. So it's just telling you the structure of the cochlea. And this membrane becomes more flexible as it widens, allows it to detect variations in sound pitch. Right, so the cochlea is going to be responsible for helping us be able to transduce sound. Right, so it supports the spiral organ of the corti within the cochlear duct. Contains receptors for hearing. Right, and here's where we convert our sound wave energy into electrical impulses inside the cochlea. Travel to the brain by axons in the cochlear division of the cranial nerve 8. So we have the vestibular division and the cochlear division, and then that comes together and we transmit all of that to the brain. So balance, orientation, head movement, balance, and then also sound. Right, so this is inside the cochlea of the inner ear. Oval window, round window. Okay. All right, so how does this work? How do we hear? All right, so similar to light, we need to sort of understand what sound is before we can really get into the explanation of how we hear. All right, so sound must be understood to truly appreciate hearing. The term hearing is audition. That's the term here. That's another technical term for hearing. Ears detect and transduce, that means convert sound waves into electrical signals that can be processed by the brain. What are sound waves? They're generated by the displacement, and this is where we get to the physics, just like when we talked about the photons and the wavelengths of light. It can get a little confusing. This is not a physics class, so I don't expect you to memorize everything about sound, but sound waves are generated by the displacement of air molecules in response to a moving object. So it's movement of the molecules in the air, vibrations. Adjacent molecules collide with one another, transferring energy, become waves that radiate away from the object in 3 dimensions. We can describe sound in pitch as well as loudness or volume, what we would normally say. Every sound has a certain pitch or frequency. If you look at sound waves, a slow frequency or fast frequency, and then how loud it is, the volume, the loudness. So the frequency, the pitch is determined by frequency. Higher the frequency, typically the higher the pitch. So determine how many times the object vibrates back and forth. That's frequency measured in hertz. Human ears can typically detect sounds between 20 hertz or 20,000 hertz. That's just like we can only see in a certain wavelength of light, we can only hear a certain frequency of sound. Too low of a frequency, we can't hear it. Too high of a frequency, we can't hear it. Notice, I think with the dog whistles, I think the frequency is too high for us to hear. Loudness or amplitude of sound, determined by a difference between areas of high and low pressure, measured in decibels. The greater force placed on an object causes greater difference between waves of high and low air pressure. So if I hit something soft, or I hit it hard, it makes a louder sound. All right, and this is just showing you some of the basic principles of sound. Low frequency versus high frequency changes the pitch. Low amplitude versus high amplitude. So these are technically the same frequency, four waves, but this is a higher amplitude this way. Therefore, it's going to be a louder sound. All right, so let's get back to the ears. So sound waves must be funneled into the ear. Sound waves cause the membrane to vibrate at the same frequency. Right, so sound funnels into your ear, and you have the ears start, the vibration happens at the same frequency. Vibration of your membrane, of your tympanic membrane. So the tympanic membrane will vibrate at the same frequency of the sound that you're hearing, and vibration of that membrane is transferred to the ossicles, those three bones in the middle ear, and transferred to the oval window into the inner ear. Vibration of the oval window generates waves in the fluid in the inner ear, mostly the perilymph and the vestibule and scaly vestibule. All right. Ossicles act as a lever system, amplify the vibration differences between larger tympanic membrane and smaller oval window to compensate for transfer of sound. Right, and that's how we get sound from the outside to the inside of our ear. Right, so how is that sound processed? We see that sound causes vibration of our tympanic membrane. Now, tympanic membrane, I didn't mention this earlier, but some people call it the eardrum. That's the same thing, right? So it causes vibration of our tympanic membrane, then our ossicles, and then the fluid in our inner ear. Right? Now that we know how sound gets into our inner ear, how do we convert that into an electrical signal that can be processed by our brain? Pressure waves generated at the oval window are transferred to the perilymph. Remember, that's a fluid of the vestibule. Then the scala vestibuli of the cochlea. So high frequency vibrations take a shortcut through the cochlear duct to reach the scala tympani, cause endolymph and basilar membrane to vibrate. Vibrations occur close to the base of the cochlea where basilar membrane is narrow and stiff. Low frequency vibrations, the bass, when you're listening to music, the bass, low frequency vibrations or sound waves travel further into the cochlea where the basilar membrane is wider and more flexible. So these low frequency sound waves are heard basically transduced by a different part of your ear than the high frequency. Now, very low frequency sound waves that are outside of our range of hearing travel to the tip of the cochlea where the scala vestibuli is connected to the scala tympani at the helicotrema. All these words I wonder sometimes, where do people come up with these names? But these sound waves can travel back to the vestibule, cause round window to vibrate. Round window is essential for allowing fluid to move within our ear. Right, so very low frequency sound waves can allow fluid to just move within our ear. Right, so that's showing you the sort of the structure. So the structures responsible for processing sound waves in our ear. Okay, so we hear a sound waves come in our ear, it vibrates our eardrum or tympanic membrane, and it's connected to the auditory ossicles, the three bones here, cause oval window to vibrate at the same frequency. Oval window here produces Vibration of the old produces pressure waves in the perilymph and scala vestibuli and the endolymph of the cochlear duct, which then vibrate the basilar membrane. Showing you a picture of that here, basilar membrane. The high frequency sound waves travel only a short distance into the cochlea, cause the basilar membrane to vibrate where it is narrow and stiff. But the low frequency travel farther into the cochlea and cause the basilar membrane to vibrate where it is wide and flexible. Okay, so and then come see it coming out here, out of here at the round window. So we have pitch, determined by vibrations per second, the frequency. All right, so it's determined by which area of the basilar membrane vibrates. Loudness, your volume, your decibels, determined by how much of the basilar membrane vibrates. All right, so more of the membrane will vibrate if the sound is louder. The spiral organ rests on the basilar membrane, allows for transfer of sound wave energy to region where vibrations can be converted, transduced into electrical signals. Spiral organ. All right, now we're getting into the transducing part of it, converting the sound into a signal. All right, so it has hair cells, receptor cells within the spiral organ grouped into inner and outer hair cells. Inner hair cells form a single row of cells. They detect sound. Now don't think of, when we say hair cells, we're not necessarily talking about the hair, like the hair on your head. Each hair cell is equipped with microvilli called stereocilia. Stereo, think of stereo, music, sound, cilia. Remember cilia, we talked about that, projections, and extend from the cytoplasm, from the plasma membrane, extend from the cell into the endolymph of the cochlear duct. The stereocilia vary in length. They're arranged from the longest to the shortest, flex at their junction with hair cell body. All right, so we have, this is a inner hair cell. Now you can't, now the hair like on my face is not the hair we're talking about. That obviously that hair does not hear. This is a specialized, what we call a hair cell of the spiral organ. Outer hair cells here have tectorial membrane, outer hair cell with the outer hair cells with the stereocilia here, basilar membrane. This is all part of the spiral organ. Remember, the basilar membrane is vibrating. All right, so that tectorial membrane that we talked about is a stiff structure, extends over the hair cells, sandwiches the cells in their stereocilia between tectorial membrane and basilar membrane. The stereocilia on the outer hair cells are in contact with the tectorial membrane. Sorry, my kids are downstairs playing video games and not being too quiet right now. So speaking of, I can hear them while I'm trying to teach this lesson. Cool thing happens is the tallest stereocilia bend as they bump into the tectorial membrane, causes all the linked shorter stereocilia to bend in the same direction. Right? So tallest one, and then they're linked here. So when the tallest one starts to bend, it causes the other ones to bend of the stereocilia. Right? And it's linked to this tectorial membrane, all part of the spiral organ. Right? Notice the nerve fiber of the cochlear nerve is down here. So when we sense, when this is starting to bend because of the vibration, it's causing synaptic vesicles and sending a signal. So converting the vibration that feels here at the cilia to a signal that is sent to the nervous system. So sequence of events: when sound is transmitted, basilar membrane moves up toward tectorial membrane, bending your stereocilia toward Talus stereocilium. Bending stereocilia opens potassium ion channels, depolarizes this specialized hair cell. Potassium ion concentration in endolymph is high enough to cause ions to flow into and depolarize the hair cells. This is a specialized structure, this hair cell. Polarized hair cell releases neurotransmitter, triggering action potentials. And that action potential is then sent down. So without further ado, this depolarized hair cell, the specialized cell that helps transduce sound waves into an electrical signal that can then be transferred to the nervous system, triggers an action potential in the axon of the cochlear division of the cranial nerve number 8. Right, so that is the key. So that basilar membrane vibrates, moves up toward the tectorial membrane, bending the stereocilia. This bending of the stereocilia causes the potassium channels to open, depolarizes the hair cell, the specialized cell, which then causes the polarized hair cell releases neurotransmitters and causes an action potential to travel into the cochlear region of the vestibulocochlear cranial nerve number 8. Right, that's how we hear. So the auditory pathway is electrical impulses transduced from the sound waves travel along the axons of the nervous system, along the pathway. So it travels to the axons of the cochlear portion of the vestibulocochlear nerve, so that's cranial nerve number 8, to the cochlear nuclei. Axons in the cochlear nuclei contact the superior olivary nucleus in the pons, that's in your brainstem. Information from both ears is compared for determining the location of incoming sound. Auditory stimuli then sent to the inferior colliculus of the midbrain, which is involved in the startle reflex. We get startled, we jump. Auditory stimuli are relayed to the medial geniculate nucleus of the thalamus. Remember, the thalamus is sort of like our control center. Thalamus sends the signal to the primary auditory cortex in your cerebral cortex, superior portion of the temporal lobe. Temporal lobe, superior portion. The primary auditory cortex is where we become conscious of incoming sound. So that's the part of our brain that really starts to realize, okay, where is the sound coming from? Is it a high sound, a low sound, a loud sound, a soft sound? That's in your cerebral cortex, your primary auditory cortex of the cerebral cortex. Primary auditory cortex is connected with other regions of the temporal lobe involved in language and limbic system. So sounds can trigger certain emotions. You can hear a song. You hear a song. I was just listening to Janet Jackson, Anytime, Any Place. The other day, it came on the radio. I hadn't heard it in, probably felt like I hadn't heard it in a couple years. But as soon as that song came on, I remembered certain moments, certain people or person. So it's tied to certain emotions and certain memories. Right, so this is the pathway of sound. From the ear to the brainstem to the thalamus to the cerebral cortex, temporal lobe. Okay. You can read through these slides. Tinnitus, when you're hearing a sound that's not actually there. It's due to a damaged stereocilia of the cochlear hair cells. It can cause you to hear sounds that aren't really happening because you're getting sort of these specialized hair cells are releasing neurotransmitters and making you think that you heard a sound that you didn't hear. There's ways around it. Sometimes people use antidepressant medications. Sometimes people use white noise to help block out sounds. So kind of an interesting situation. We also have hearing loss. Now, this is something that a lot of people have experienced, and it can be caused by different reasons. It could be a problem due to the outer or the middle ear, which prevents sound waves from reaching the inner ear, buildup of earwax and ear infection. You ruptured your tympanic membrane, all can cause hearing loss. Sensory neural hearing loss, you can have an issue with your cochlea or any part of the neural pathway. If the nerves can't transmit, even if your ear is perfectly fine, but if there's something wrong with the way the nerves, the vestibular cochlear nerves transmit that sound, you won't hear it. So even though you have the vibration in your ear, if it's not transmitted to the brain, you can't hear it. Okay. You can do such things as cochlear implants, surgically implanted electrodes, can bypass damaged hair cells and directly stimulate the cochlear nerve. There's ways to do that. Hearing implants, a lot of people have these and these are very successful. Help you maybe to hear sounds that you would not normally be able to hear. And you can read about that here if you would like to read more about that. Right, and then there's a bigger, showing you the big picture of hearing. And you can look at this chart and analyze it for the detail, sort of get the big idea. But remember, hearing, basically what we want to do is the sound waves, which are a certain frequency, certain decibel, that they want to be channeled into the ear, vibrate our eardrum, our tympanic membrane, vibrate the ossicles, vibrate the fluid in our cochlea, and be transduced by those specialized hair cells into a nerve signal, into an electrical signal that can be transmitted to our brain, through our brainstem, thalamus, and to our cerebral cortex, primary auditory cortex, and then we can understand that this is what I am hearing. Now, we also have the vestibular sensation, which is also associated with the ear. Right, so it's a sense of equilibrium, depends on input from our sight, proprioceptors in the muscles and joints, and the vestibular system. The vestibular system includes your vestibule, semicircular canals within your ear, which we talked about, which provide information about head position and movement. It's known as the vestibular sensation, right? So balance, we're talking about balance here, sense of equilibrium, balance. So it's a combination of strength, muscles and joints, your sight, something. Notice it's harder to balance if you're balancing on one foot. They say stare at a dot on the floor, for instance. It'll help you balance, because if you balance on one foot and then you try to look around side to side, you'll notice that it's harder to stay on balance. So all these systems work together. So the inner ear contains organs that detect two types of equilibrium. You have the static equilibrium, maintaining balance when head is tilted, but head and body are not moving. And you have dynamic equilibrium, maintaining balance when head or body is undergoing rotational or angular motions, or linear acceleration, such as riding in a car. All right, so your body try to help, this vestibular sensation tries to help balance your body out, whether your head is tilted, but the body's not moving, or whether your head is moving. It's being your body, you're being rotated, or you're moving in a car, for instance. All right, so let's look into the utricle and saccule segments of the membranous labyrinth in the vestibule. Both contain a macula. Macula is receptor cells for head position and movement. We wanted, and remember, just like sound was transduced into the cochlear region of the vestibule, cochlear nerve, cranial nerve number 8. This is also sending. This is the other half of that nerve, and your body is sending information through that nerve as well, about your vestibular sensation. So not only are you getting sound information, but you're getting balance, equilibrium information as well, all in cranial nerve number 8. All right, so the macula contains receptor cells for head position and movement. Each hair cell displays stereocilia and one true cilium, taller than the stereocilia. Kinocilium and stereocilia are embedded in a gelatinous mass, suspended in endolymph. Otoliths, calcium carbonate crystals, and otolithic membrane increase membrane density. These hair cells release neurotransmitter glutamate continuously onto dendrites of bipolar neurons that form vestibular division of your cranial nerve number 8. Bending stereocilia alters release of inhibitory neurotransmitter glutamate, alters activity of vestibular neurons. So they're constantly, so normally these hair cells in this region are releasing the neurotransmitter glutamate continuously, basically telling your body, I'm balanced, I'm balanced, I'm balanced. But when you have a bending of the stereocilia, it alters the release of this inhibitory neurotransmitter glutamate, alters activity of the vestibular neurons. So there's only a problem when you have a bending, when you have something that takes you off your normal, knocks you off your square, so to speak. So stereocilia and utricle are vertically oriented when head is upright and stationary. So I'm upright, stationary, everything's cool. So there, it's basically releasing the neurotransmitter, everything, no signal, it's releasing the neurotransmitter, which inhibits any type of notification that I'm off balance. They bend in response to tilting head to the side to side, or front or back. So stereocilia and saccule, and the stereocilia and the saccule are horizontally oriented when head is upright and stationary, and they bend in response to up and down movements of the head. Okay, so side to side, the stereocilia in my utricle will be bent. All right, up and down movements of the head, the stereocilia in my saccule will be bent and send a signal. All right, it'll stop sending the signal that everything is okay. All right, so this is in the saccule, utricle, and saccule, semicircular ducts, maculae, cochlea. All right, so when the head tilts, gravity pulls on the otolithic membrane, bends your stereocilia. Stereocilia bend toward kinocilium, hair cells depolarize, release more glutamate, increased number of action potentials produced in the vestibular nerve. When they bend away from kinocilium, hair cells hyperpolarize, release less glutamate, reduces number of action potentials. All right, so when they go back. All right, so it's kind of showing you what's happening here. So when your head is straight, they're nice and straight, everything is fine. You bend the head to the left, for instance, it increases. When you come back to center, everything's fine. Bend the head to the right, these will bend, you have more action potential. Sorry, bend your head to the right, you'll have even less. So in the middle is the normal. Remember we said it's continually releasing glutamate. So it's normal in the middle. Bend your head this way, it increases. This way, it decreases below normal. And that's how we know something is, we're sort of off balance. The maculae are paired so that tilting your head in one direction polarizes hair cells in one ear, hyperpolarizes hair cells in the other, allows the brain to determine which direction head is tilting, right? So you're based on how your head tilts, it polarizes one side versus the other side. So we can, even with your eyes closed, you can tell which way your head is tilting. They're also involved in detecting linear acceleration. Maculae are sensitive only to changes in rate of movement, not movement itself. So acceleration, as you speed up, you feel it. You feel like the pressure, it just draws you back. They're sensitive to changes in, or slowing down, for instance, changes in your speed. So imagine you're stopped at a red light, riding in the front passenger seat of a car with a cup of coffee. Light turns green, driver stomps on the gas pedal. Why are you now wearing coffee on your shirt? Because that coffee spilled on you because its car sped up too fast. That's similar to what's happening with your endolymph and otolithic membrane, all right? Movement lags behind that of the hair cells and the walls of the utricle and saccule, bends the stereocilia, and you can feel that movement. Your driver slams on the brakes, the reverse happens. You sort of go forward, the fluid in your ear moves forward, you feel it. You know that you're moving forward. When the hair cells and the rest of the body stop, the endolymph and the otolithic membrane continue to move forward, bend the hair cells in the opposite direction. That when cars are accelerated, right? You feel that force. In other words, if nerve activity increased when you first start moving, it will now decrease. Brain interprets as slowing down or decelerating. Right? So the same thing. So very similar to a coffee cup. How you when a car accelerates, the fluid in the coffee cup doesn't accelerate as fast as the rest of the car. Therefore, if it accelerates too fast, the fluid will spill on your shirt. And if it stops, when the car stops, the fluid doesn't stop as quickly, so the fluid would spill out of the front of the cup. And so the same fluid is moving. We have similar idea. The fluid is moving in our semicircular canal in our ears. And our endolymph is moving otolithic membrane, and that's how we can tell when we're slowing down or speeding up. These semicircular ducts, hair cells, and anterior, posterior, and lateral semicircular ducts of membranous labyrinth detect changes in dynamic equilibrium, angular rotational movements of the head. They're oriented to detect rotation in all three body planes, and they contain endolymph, the same fluid. Each duct is connected to utricle at enlarged region called the ampulla. This is that structure here. All right, at the ampulla, these semicircular ducts are connected to the utricle. You can detect movement. Each ampulla contains crista ampullaris, cluster of hair cells, stereocilia, solitary kinocilium are embedded in gelatinous mass. Similar to the maculae, head is turned, endolymph lags behind, pushes on the cupula. They bend in one direction, increasing or decreasing glutamate release from the hair cell. They bend in one direction, it increases glutamate release. Bend in the other direction, decreases it. But remember, normally, if everything is straight, your body is just releasing a constant amount of glutamate. We only change it if there's a bending in one direction or the other. Changes in the glutamate release causes changes in the neurons of the vestibular nerve. Brain interprets that change just like it does with the saccule and the saccule. It interprets that change, that movement of those of these hair cells. When head stops moving, endolymph continues to move, bending the cupula in the direction opposite the direction head was moving. Movement has opposite effect on glutamate release and activity of vestibular nerve. Signals head has stopped moving, right? So same idea, detects movement. Oops. All right, so this is more rotation. So rotation. Endolymph, that's the fluid, cupula, stereocilium. Those are this part of the hair cell that helps transduce movement into a nervous signal. And based on if the head is turning one direction, increases glutamate release, head stops turning, decreases glutamate release. Below normal, this is above normal glutamate release. This is below normal glutamate release. And it can send a signal to your nervous system that this is happening. You may have heard of motion sickness. Sometimes people get nausea, they throw up, they get dizzy. Brain receives mismatched sensory information from the eyes and the vestibular system. So this happens oftentimes in a car or a train or a plane. You'll see something moving beside you, so it looks like something is, through your peripheral vision, it looks like something's moving, but then you're trying to read a book, for instance, which is stationary. So your brain says, I'm moving on one hand, but I'm stationary on the other, and it gets mismatched signals, and we get what we call motion sickness. Right? That's basically what's happening. Brain cannot integrate contradictory information, causes symptoms of motion sickness. And so that's why some people can't read on trains or on moving objects. Okay. All right, so what is the pathway, the vestibular pathway? So brain must receive and analyze stimuli from vestibular input, coordinate appropriate response. So responses to the vestibular input can be grouped into the following categories: muscle movement, cognitive awareness, we notice it, compensatory eye movements for some types of head movement. Vestibular signals must reach the following regions of the CNS to produce appropriate responses. So your vestibular signal travels to the vestibular nuclei located in the medulla pons junction in your brainstem. Vestibular nuclei forward signals simultaneously to the thalamus, then to the inferior parietal lobe. That's the part of the brain. Conscious awareness of head position and movement. We also have the cranial nerve nuclei associated with extrinsic eye muscles, coordinate eye movement in response to head movement. Your cerebellum and your spinal cord coordinate muscle movement that maintains balance in response to head movement. So if I move my head, or if I tilt my head and I need to, I can balance my body. That's why usually if you're trying to balance like on one foot, for instance, it's better to focus on one spot that's not moving. It'll help you with your balance. Otherwise, your brain will try to respond and help you. If you're moving, it'll help you try to move your muscles to try to balance. And this is a diagram of that pathway.